All right, now let's talk about phagocytosis. Phagocytosis is an important process, so let me write it down, phagocytosis. And the phagocytosis is the defense mechanism of the innate immune system. So it's a defense mechanism and really important part of the innate immunity. And using this, bacteria basically get destroyed by the immune cells. So let me show you by using the diagram, and let's say this is the immune cell, right? And it has got receptor over here. So this is the immune cell. And whenever a particular pathogen or invader, something that I've created in red round structure, is getting recognized by the immune cell, and it will be now engulfed or eaten up and then will be destroyed. So three important components are going to be there, and this is the invader, right? So the immune cell will basically recognize, will phagocytize, eat it up and destroy. So it is made up of different words. First one is phagin, means to eat, right? And the second part is cyte, which is cell. So it's coming from the different words over here, and osis, phagocytosis, right? So I'm just breaking it down, and that is process. So essentially phagocytosis is cell eating process, because this is the literal meaning of phagocytosis. So what happens is basically it's an earlier response, immune response. Whenever there is invader, the first response will be recognizing and then phagocytizing and eating up the invader. So it comes before the adaptive immune response. Adaptive is specific immune response. That's why it's a part of the non-specific immune response. Now let's talk about the major phagocytic cells. Let me write it down. Phagocytic cells. The first one is neutrophils. I hope you remember from our last discussion also, neutrophils. And what they do is they rapidly get recruited to the site of the entry of the pathogen, and then further they are highly efficient also. Okay, next is they produce a lot of antimicrobial granules, right? Let me write it down, granules. And the fourth feature of these neutrophils is they are short-lived cells. They are not very long-lived cells. They are short-lived cells. These are the main component. Let's talk about second cell, which is macrophages. Macrophages basically are really long-lived cells, and they are derived from the monocytes. Monocytes, essentially, they get converted into macrophages. Their role is basically to remove the pathogen, and also, let me write pathogen, and also the dead cells. And then next important role of these cells is cytokine production, those molecules which are basically activating the immune response, right? And then let's talk about the third cell, which is dendritic cell. All these three cells, they play a very important role in phagocytosis. And they are, dendritic cell, they are APCs, antigen presenting cells. They activate T cells. Dendritic cells are really important in activating T cells. And that's why they are a kind of a bridge between the innate and the adaptive immune response. So dendritic cells are the cells which actually make a bridge between these two types of immune responses. Now let's talk about the stages of phagocytosis, right? What are the different stages? And if I show this particular, these stages by using illustration, so let me create this. Let's assume this is an immune cell. And right over here, receptor is going to interact with the pathogen. And this is, let's assume this is a pathogen, and its surface antigen is interacting with the receptor, which is present on the cell. So that will be first step. Second step will be the downstream signaling pathway. So a lot of different proteins, blue protein, red protein is getting activated. That will be the second step, where cellular signaling will be activated, and then further what is going to happen is basically it's going to get entered inside that particular cell, and you will have that phagosome which is getting created inside the cell, right? So this is the phagosome. Let me label it down, phagosome. And that phagosome, further inside that, the lysosome will deliver its content, and that inside that the killing will happen of that particular pathogen. So it's going to get killed, and then the part is going to get processed on the surface of the cell If those are the antigen presenting cells, so they will be doing antigen processing. If there are other cells, they will be they will perform exocytosis. So first step, if I write it down, is recognition that I told you earlier. Second one is signaling, downstream signaling. Third for phagocytosis is attachment or engulfment. So bacteria will or the pathogen will enter. Fourth is phagosome formation, and then killing is the next one. And the last one is antigen processing or exocytosis if the entire component is going to go out, right? These are the important stages of phagocytosis, okay? Now let's talk about the first one, which is recognition, first stage. So now we are diving deep into the phagocytosis. So in recognition, what will happen? You have immune cell. It's going to interact with this this pathogen. Let's say this is the pathogen, and this is going to recognize this as foreign molecule, right? As a foreign molecule, outsider molecule. And how this recognition is going to happen? It's going to happen with pattern recognition receptors. They are also known as PRR, pattern recognition receptors. In bracket, PRRs. Now, PRR will detect molecules called PAMPs, PAMPs, pathogen-associated molecular patterns. So PRRs are basically microbial structures. They will detect microbial structures. PAMPs are the microbial structures, right? So these are let's assume this is the bacteria, and you can see flagella is there. You can see surface molecules are there. All these are PAMPs. These are PAMPs. And PRRs are the receptors that are present on the immune cells. So microbial molecules are called the PAMPs, and then they are basically absent in the host cell. They are not present in the host. That's why the immune response is generated, because they are treated as non-self. They are going to get treated as non-self. And if we talk about the example of those molecules, let me write it down. Examples. First one is LPS, lipopolysaccharide. Second one is peptidoglycan. These are cell wall components of gram-negative and gram-positive bacteria. Bacterial lipoproteins, okay, bacterial lipoproteins. You have nucleic acids like DNA, RNA that will be detected. You have fungal glycans, especially beta glycans. Let me write it down, beta glycans. These are examples of all the important PAMPs. Now if we talk about second category, which is pattern recognition receptors, as I already told you that these are the receptors present on the immune cells, and they will interact with PAMPs. The examples, let's talk about these examples of PRRs. First one is TLRs, also known as toll-like receptors. Okay, if I diagrammatically try to show you, let's suppose this is the surface of the immune cell, and this is the structure called TLR. It will have these are basically membrane receptors, right? And they will have extracellular domain. They will also have intracellular domain. So let me write it down. This one, which I'm showing, is extracellular domain, and they will also have endosomal TLRs also there. So you'll have extracellular TLRs, endosomal TLRs, right? The important TLRs, let's talk about them. First one is TLR4, very, very important. It detects LPS, lipopolysaccharide. TLR2, this detects peptidoglycan molecules, and it also detects lipoproteins, right? So it becomes very, very important for us to know what are TLRs. TLR9 is basically unmethylated CpG DNA, okay? CpG DNA motifs, and they are basically coming from bacteria. So TLR9 will detect the DNA of the bacteria, right? Now let's little bit dive deep into the TLR signaling pathway. So how the signaling happens, how TLR is able to recognize these molecules. So TLR signaling pathway. Let's suppose this is the surface of the cell, and on the surface you can see this is the TLR. TLR will bind with its ligand molecule that we discussed earlier. So it will binding will happen, and then there will be downstream signaling pathway. The first one is MID88 signaling pathway, MID88 signaling pathway. In this one, the protein which is MID88, and the full form of this protein is myeloid differentiation primary response 88. This is the name of the protein that gets activated, and further you have multiple proteins that are going to get activated by this particular protein. You have IREC, you have TRAF6, you have DAK1. All these proteins are going to further activate the protein which is NF-κB, nuclear factor κB. That activation is really important. And then this, when this gets activated, further gene expression is going to happen, right? So this is going to trigger immune response, a very, very strong immune response. So NF-κB is usually in the resting stage. It's usually the inactive form of this particular protein. Why? Because with this particular protein, there is another protein which is attached, which is IKB. It's inhibitor. So if that protein is attached, it's going to inhibit. But if degradation of that protein happens, NF-κB moves inside the nucleus. So it's going to move inside the nucleus, and then it's going to trigger the immune response. And then a lot of different molecules or genes, they are going to get expressed. So this is how entire TLR pathway will occur. Now let's talk about the next one, which is the genes activated by NF-κB. This is really important. So what are the genes that are activated? One is TNF-alpha, IL-1 beta, IL-6, and then various chemokines, as well as co-stimulatory molecules. Co-stimulatory molecules. They are getting activated when TLR pathway gets activated. Functions. Let's talk about functions. Like TNF-alpha, this is really important. It increases the inflammatory process. So it's a pro-inflammatory cytokine. It enhances the inflammation. Also, vesicular permeability is enhanced. So whenever there is TNF-alpha release, it's going to cause high inflammation. IL-1 beta, which also increases fever and also inflammation. So it's also pro-inflammatory cytokines, right? IL-1 beta. Then you have IL-6, which is acute phase protein. IL-6, acute phase protein. Then you have next one, let me write it down with a different color, IL-8. It's also known as CXCL8. And this helps getting the neutrophils at the site of the infection. So neutrophil recruitment at the site of the infection is basically done with IL-8. So it's really, really important, all these cytokines and their functions. Now, let's move on to the next phase, which is attachment in phagocytosis. That is what we are understanding. Attachment and engulfment. How the attachment will happen and then how the bacteria will enter inside the cell. Let's assume this is immune cell. First, it will interact with the antigen, and then it's going to get entered specifically by using membrane folding. So just imagine this is how the membrane is going to get folded. This is membrane. These are membrane projections, and also they are known as pseudopodia, false feet. So bacteria is going to get trapped by these membrane projections. So actin cytoskeleton rearrangement is a very, very important process for this particular pseudopodia formation. Further, there are various proteins that are involved. The first one is RAC protein. Second one is CDC42 protein. And then you have Rho A. These are really important proteins that are involved in this. Actin polymerization. Okay, their role as membrane movement. So membrane movement will also is required for the eating process. Pseudopodia formation, false food formation. False food formation will happen because of these proteins. And then another important process is opsonization. Because when there is opsonization, means attachment of antibodies or complements on the surface of the pathogen, it will cause enhancement in the phagocytosis. This is opsonization. And it will have, it will enhance the phagocytosis process. So it will have antibodies. These are Y-shaped structures that are going to bind to the pathogen. Complement, specifically C3b complement, okay? That get coated on the pathogen, and that will cause the enhancement in the phagocytosis. Now let's talk about the receptors. One of the important receptor is Fc gamma receptor, and Fc gamma receptor is basically present on the immune cell. So if I draw, let me show diagrammatically. This is the pathogen, and antibody is bound to this pathogen, and this is the receptor which is present on the on the cell, and this is the Fc gamma receptor, which basically bind with the Fc fragment of the antibody. Because of this, what will happen? Enhancement in the phagocytosis will occur. So another receptor is CR1, which is recognizing C3b coated pathogen. So for complement, you have CR1. For antibody, you have Fc gamma. Now CR3 is another one which which helps in enhancement of the internalization of microbes. So another receptor is there, which is involved in entering or moving the pathogen inside the cell. So let's suppose this is a pathogen, right? These are the antigenic molecules, and antibodies are binding. Complement is binding. Blue color is complement, white color is antibody, and here is the receptor, you can see. And then right over here, another receptor, that is the complement receptor. So right over here, this one is Fc gamma receptor, and this one is complement receptor, which is CR1 in this case. I'm just giving you an example, right? This is how opsonization will happen, and the bacteria will enter inside the cell. Okay, now let's talk about phagosome formation. Really important. Now we have talked about the attachment, now phagosome formation. Phagosome is nothing but the vesicular structure that will be present inside the cell. So let's suppose this is the pathogen and the blue colored circle is the phagosome. This is the phagosome. Initially, the phagosome role is early sorting compartment, right? It's just involved in sorting various molecules. So it's early sorting compartment, and it needs maturation. Maturation is really important for proper functioning of the phagosome, where degradation will happen. So maturation, what happens in maturation? Let's talk about that. In maturation, what you'll have is phagosome. So let's suppose this is the phagosome, and this is lysosome. Let me label them. So what will happen? They will basically get fused together. So that's the maturation process, and lysosome will bring its all component inside the phagosome, right? So the vesicle formation will happen. Okay, so what basically will happen? Various proteins are going to get involved. One is Rab5 and Rab7. These are two important proteins that are involved in vesicle formation or maturation. Rab5 is involved in early phagosome formation, right? And the Rab7 is the late phagosome indicator. So basically, transition from Rab5 to Rab7 will indicate the maturation. Then you have snare proteins. They will be involved in membrane fusion, or they will cause the fusion of phagosome with plus sign lysosome. Snare proteins are involved in that one, right? So this is your phagosome, and the blue one right over here is the lysosome. So this is how the phagosome formation will occur. This is how the maturation will occur, and then snare proteins are the proteins that are going to cause that fusion of these two structures together. Okay. All right. Now let's talk about the next process, which is killing and digestion. Killing and digestion. Let me write it down right over here. So how the killing will happen, that is really important. So killing as well as complete digestion of that particular pathogen, how that will happen. So you have molecules inside of membrane, which is lysosome. So molecule is lysozyme basically. And the role of this lysozyme is nothing but degradation of peptidoglycan. So target is peptidoglycan, which is present in the cell wall of the bacteria. So lysozyme is present inside the lysosome. So you have proteases also. These are the enzymes that will degrade proteins. Let me write it down. Degrade proteins. You have you have enzymes that can degrade DNA. So DNases, RNases will be there. These enzymes can degrade the genetic material. Then you have phospholipase A2, very, very important enzyme that can lyse membranes, right? And then further, another important process that happens is acidification of the phagolysosome. When there is the fusion of phagosome with lysosome, it becomes phagolysosome. And then pH will be significantly dropped inside that particular structure. So let's assume that this is the phagolysosome, and you have vacuolar proton pumps. These proton pumps, they play a very, very important role in lowering down the pH and making the pH acidic, right? And because of that, these enzymes, they work really, really fast, high activity or high killing. Activity and killing will happen, and those pathogens, red color pathogens, they will get immediately degraded. So that is very, very important for digestion of these molecules. Okay. Now let's talk about how the death happens. Molecular mechanism is basically related to respiratory burst. What happens is there is a sudden increase in O2 consumption. Cell will consume oxygen very, very fast. So that is the respiratory burst process. What happens is it basically produces toxic oxygen intermediate, or they are also known as reactive oxygen intermediates, ROIs. These molecules, they are really antimicrobial in nature. So the protein which is NADPH oxidase complex, which is also known as NOX2, plays very important role in that process. You have GP91 FOX protein. You have P22 FOX. All these proteins are the part of this particular complex. You have P47 FOX. You have P67 FOX. You have P67 FOX. I'm writing again, right? So you have P40 FOX, and then RAC GPTase. All these proteins, they are really, really important. And these are basically cytosolic part of this particular complex. And the top one, they are extracellular. Now you have, further the important part is superoxide radical generation will happen because of this particular complex. And then what it does is basically kill the microbes. It will damage the microbes. And the component of those radicals, the specific radicals, they are, you have O2 negative, you have H2O2, you have OH radical, you have singlet oxygen. All these molecules, they basically play a very important role in killing the microbes. All right, now let's talk about another important system, which is myeloperoxidase system. Myeloperoxidase system. This system is basically nothing but an enzyme which is produced by neutrophils. In short, it is also known as MPO. The role of this enzyme is to produce HOCl, which is hypochlorous acid. And this hypochlorous acid is very, very potent biological bleach, highly potent antimicrobial compound. It is highly antimicrobial in nature. So let me write it, antimicrobial in nature. Another component is RNI. By using all these components, basically pathogen gets killed. And it is basically produced by activated macrophages, and in short known as reactive nitrogen intermediates. Example is NO, which is nitric oxide. Nitric oxide is another antimicrobial agent. Then you have INOS. INOS is basically it's going to produce its inducible nitric oxide synthase. It's going to produce NO. So NO is going to get produced by INOS, and by using amino acid arginine. Okay, so arginine will be used, and then NO will be produced by this INOS. Okay, what else? You also have, let's talk about function of NO, which is nitric oxide. It's antimicrobial in nature, but we need to know the functions. The first one is it works on microbial respiration. So it inhibits microbial respiration process. Basically targets the electron transport system, ETS system. That's why bacteria or pathogen dies. Next is DNA and also various enzymes. They are targeted by this, and it also causes tumor cell killing. Next is peroxynitrite formation. Next is peroxynitrite formation. So this is again very, very important component of innate immunity. You have NO, nitric oxide, plus superoxide radicals. When they combine together, they form peroxynitrite, which is again extremely antimicrobial in nature. Let me write it down. Extremely antimicrobial, extremely antimicrobial. See, if I show you with illustration, let's suppose this is the foreign invader. This is the bacteria. I'll use red color for that. I'll create an illustration to show you that this is the bacteria. Okay, let me create that. This is the red colored bacteria. This is the DNA. And then inside that you have various proteins that are functioning, critical processes in the cell. So function of proteins, it's going to get targeted with NO. It's also going to target the membrane. So membrane lysis will happen. It's also going to target the DNA. So all these targets are the targets of NO. So it's going to basically immediately kill the bacteria. Next component is antimicrobial peptides. So immune cells will also have antimicrobial peptides. Antimicrobial peptides, they are also known as defensins. Some of the important antimicrobial peptides are defensins, right? These defensins, they are produced by neutrophils. So let's suppose this is neutrophils. Inside neutrophils, you have granules, and these granules contains defensins. This is nothing but antimicrobial peptide. Let me write it down, antimicrobial peptide. when produced by neutrophils. Let's label it neutrophils. These are the neutrophils that are going to produce these compounds. Now, if you talk about the mechanism, right? Mechanism of action of defensins. So suppose this is the bacterial cell, you have DNA inside. So defensins, what they will do, they will create pores in the bacterial cell wall. So formation of pores will happen. Further, they can, the component will leach out. Leakage of the component will happen. They will target gram-positive bacteria. They can target gram-negative bacteria. They can target fungus. They can target viruses, especially the enveloped viruses, because they mostly affect the membrane of these pathogens, right? Another one is called netosis. Really interesting term. Netosis is basically advanced neutrophil defense system. And what it contains? It contains, extracellular traps, and also known as nets, extracellular nets, which is composed with DNA, okay, histones, myeloperoxidase, that the enzyme we already discussed. You have elastase. All these components are basically the part of netosis, where the bacteria or the pathogen is going to get trapped. So I'm just creating illustration for trapping this particular bacteria with these lines, and then to trap the microbes, right? And so that antimicrobial peptides can now work on these microbes. If they keep on moving, then it's very difficult. So antimicrobial peptides, they can basically target this trapped bacteria. Okay. So now let's move on to the next part, which is inflammasome activation. Inflammasome activation. Let me write it down. Inflammasome activation. As the name indicates, it's related to inflammation, right? So you have cell, let's say this is the cell, and inside bacteria, you have bacteria can cause the formation of inflammasome inside the immune cell. The proteins that are important are NLRP3 protein and, ASC adapter protein, that is another protein. Caspase 1 is really important protein in case of inflammasome formation. So this is the protein. And the role of this particular protein, caspase 1, is basically to change inactivated cytokines into activated cytokines. And inactivated cytokines example is pro-IL beta, IL-1 beta. You have activated IL-1 beta. You have inactivated, which is pro-IL-18, and that will be converted into IL-18, which is activated form. And because of that, inflammation is going to get increased in that particular region, right? So that is why the inflammasome is the term for this particular process, right? So you have those vesicles where high inflammation will occur. Now let's move on to the next process, which is antigen processing and presentation. So how the antigen is going to, now the bacteria got destroyed, pathogen got destroyed inside the cell. So let's say this is the immune cell, this is the bacteria, it's getting destroyed, various components are going to get released, then receptors will be there that are going to basically activate the adaptive immunity using antigen processing, right? So what happens is, in case of neutrophils, so if I draw the structure, in case of neutrophils specifically, the presentation is not going to happen, but exocytosis, all those components are going to thrown out. So neutrophils will cause exocytosis, but antigen presentation will occur in dendritic cells and also in your macrophages. Macrophages and dendritic cells, they will cause the antigen presentation. What happens in that case is you have peptides that are formed inside the cell. They are going to express by using receptors called MSA class 2 receptors. And these class 2 receptors will activate T cells, specifically helper T cells. These are the helper T cells I'm writing over here. They are going to get activated. Plus, in addition to that, you have co-stimulatory molecules that are involved in this interaction. For example, CD80, which is B71. You have CD86, which is B72. And they are really important for T cell activation. And T cell activation is really important for cell-mediated immunity as well as adaptive immunity. Now, let's talk about some of the important processes, how pathogen evasion can occur. So pathogen can be really smart and evade this particular system. Let's talk about Mycobacterium tuberculosis, MTB. We all know tuberculosis is very, very dangerous disease. So what happens in that case is, whenever there is the formation of phagolysosome, so phagosome is going to get merged with lysosome, that fusion is required for killing the bacteria. Mycobacterium target that and there will be no fusion. So this is how it actually survives inside the cell. Next is Staphylococcus aureus. Staphylococcus aureus gram-positive bacteria produces catalase enzyme. This enzyme, catalase enzyme, that can basically neutralize your H2O2, right? If H2O2 is not there, it's not going to kill the bacteria. Next is Streptococcus pneumoniae. Very, very clever approach where this bacteria produces the outer covering, which is called capsule. This is the capsule. And the capsule, because of the capsule, the phagocytic cells, they are not going to eat the bacteria, right? They are unable to eat the bacteria. So no phagocytosis is going to take place in this case. So bacteria survives. So on the other hand, and you have immune system, on the other hand, you have smart bacteria. And that war is going on, right? And how bacteria actually evade from the immune system is very, very important. And molecular mechanism associated with that is also really important when it comes to exam and when it comes to research, right? So I hope now a lot of things are cleared. We have covered so many things, especially detailed overview of phagocytosis, right? Please now you need to read a little bit in more detail about this particular process, and then make sure that you understand this entire process. What are the components that are involved in killing the organism? What is the mechanism of the killing? What is the mechanism of recognition and all, right? Now let's move on to the next part of this particular course. And once you are done with this particular entire series of these lectures, you will have a great understanding of innate immunity. All right, let's move to the next one.